Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing the ICMR BRET question paper for the subject biological science, that is section C questions that I am discussing. This was conducted in the year 2024. So coming to the question, given below are two statements regarding ubiquitination. Binding of the carboxy terminal of ubiquitin to lysine residues of target proteins. Now, mono or polyubiquitination serve as a mechanism by which the tagged protein are targeted for destruction by the proteasome of the cell. That is correct. Okay, so the So here in a normal ubiquitination, which is targeted for degradation, what happens is the ubiquitin will be attached or bound to the lysine residue on a target protein, and the protein which is tagged with the ubiquitin, it will be targeted for destruction by the proteasome. The next statement, most ubiquitination can serve as an activating signal by which tagged proteins may form activating complex. Yes, that is also correct. Ubiquitination is not for degradation. Okay, it can be for other reasons also, such as ubiquitination can alter the protein's function by changing its shape, making it either less active or more active. Then it can act as a signal to move the protein to another part of the cell, wherever that protein is needed in that cell. Then it help a protein to interact with other proteins to perform complex and specific cellular tasks. So that is the non-degradative role of ubiquitination, and one example is in the case of DNA repair. So when a DNA damage occur, certain proteins, they are tagged with K63, that is lysine at position 63 of the protein is bound with ubiquitin chain, and this will recruit this ubiquitination. It will help to recruit other proteins which are involved in the repair process. Okay, so statement 1 is also correct, that is with regard to ubiquitin acting as a degradation, and the other one is where ubiquitin is not playing a degradation role. So the first statement, that is both 1 and 2 are correct. Then next one, the following method belong to methods to enumerate and study functional responses of T cells. So which of you have to match? So the in vitro proliferation of T lymphocytes. So to see the proliferation or division of T lymphocytes under in vitro condition, this is usually done by using the H3 labeled thymidine. So A4 is there in 3 and 4. So we will see. So A4, so it is by what is that? H3 labeled thymidine, a radioactive assay method, and here the radioactive thymidine, it will be incorporated when the cell divides during the cell divides, and during the S phase of the cell cycle, this labeled thymidine will be incorporated, and thereby you can measure the proliferation of T cells under in vitro conditions. Then in vivo proliferation of T lymphocytes, that is not in your culture medium. So for that case you will be using B2, that is 5,6-carboxy fluorescein diacetate succinimidyl ester, or CFSE. So what happens is, first these T lymphocytes, they are stained with the CFSE, which is a fluorescent dye, and during cell division the dye will be equally distributed to daughter cells. But as the daughter cells keep on dividing, the CFSE intensity will be reduced to half. Now this can be analyzed and quantified with the help of a flow cytometer. Okay. So B will be 2. So 3 is the answer. 4 is not the right answer. Okay, we will see the other options also. Then next is enumerate T cells with a single antigen specificity. So for that you use peptide MHC tetramers. So first you have to prepare this tetramer, MHC tetramer. So first you will express the MHC or major histocompatibility complex, either class 1 or class 2 in a bacterial expression system. Then you will bind this MHC with a specific peptide, and this complex then it is labeled with a fluorescence tag, and then you take the sample, that is the T cells. Okay, that is your sample. You take the T cells and mix with this MHC tetramer. Now the tetramer, they will bind specifically to the T cell receptor of the T cells that recognize this particular peptide MHC complex. Then you analyze them using the flow cytometer. Okay, that is what you do here. So C will be 3. Then to quantify cytokine secreting effector T cells. So for that you use the ELISpot method. So in this micro plate, what is that one which you used for ELISA itself, that is having multiple wells, you use that here also. Then you coat the well with an antibody that is specific for the cytokine that is produced by the T cells. Now you take the T cells and you stimulate them with a relevant antigen. Now add these T cells to the well. So those T cells that are secreting the cytokine will be bound to the specific antibody on the microtiter plate. Then after this, the washing and everything is conducted. Then you add a second antibody that is labeled with an enzyme, and it is added to detect the captured cytokine. So this secondary antibody, it will bind to the antibody which is bound to the cytokine, and upon an enzymatic reaction it will give a colored product. So and this colored product, it is visible as a spot. So each spot corresponds to the single cell that secrete this cytokine. Okay, so in that way you can do quantification also with ELISpot technique to quantify cytokine secreting effector T cell. So answer is option 3. Then before coming to the next question, we will see what is meant by positive selection and negative selection in case of T cells. So here I have shown the diagram of positive selection. So what is happening in positive selection? These are the cells which are expressing the MHC complex, and these are the T lymphocytes or thymocytes. Okay, now these those T cells that do not recognize the MHC, they will be subjected to death by apoptosis. And those T cells that recognize this MHC but binds to the MHC with only an intermediate affinity, they will be selected to continue the next stage of maturation. Okay, they will not undergo apoptosis. That is in the case of positive selection. That is, this will ensure that the T cell is able to recognize antigen presented by MHC molecule. Now comes the negative selection. So in negative selection, again a cell will present the self antigen bound to an MHC molecule. Self means your own antigen, not from any exogenous sources. Okay. So they will present the self antigen bound to an MHC to these developing or maturing thymocytes. Now, if these T cells, they recognize the MHC and self antigen with high affinity, they will undergo death by apoptosis. Okay, and those which do not recognize this MHC and self antigen, that is those which have very low affinity for binding to this MHC with the self antigen, or will not receive the death signal, and they will continue to undergo the next stage of maturation. So in positive and negative selection, high affinity cells are eliminated, whereas the intermediate cells, that is they are those cells with the intermediate affinity for MHC and MHC bound to self antigen, they are allowed to mature into functional T cells, whereas those with the low affinity, that is those that cannot bind to MHC molecule at all, they fail the positive selection and they are eliminated. So this is with regards to negative and positive selection. Now come to the question. So fewer than 5% of thymocytes produce TCRs that bind to MHC peptide complexes with high affinity. That cannot be true. Okay, they cannot bind with high affinity. MHC peptide complex binding with high affinity, they are not selected at all. Those that bind with low affinity only are selected. So they are, if it is with high affinity binding, they will not survive and mature. Okay. So it should not be high affinity, it should be somewhat intermediate binding affinity that survive and mature, not with high affinity. So statement 1 is wrong. Then statement 2, more than 90% of thymocytes produce TCRs that either do not bind to MHC or MHC peptide complexes, or bind to them with very low affinity, they die by neglect. I told you already. In a negative selection, they bind to MHC with self peptide, and if their binding is very high, with a very high affinity, then they die off. In positive selection, those that fail to bind to self MHC molecules will receive the death signal, or they undergo death by apoptosis. So the few cells survive are those with intermediate affinity. Okay, so these cells, that is which can with the intermediate affinity, can bind self MHC or MHC with peptide complexes, but not with higher affinity, but with intermediate affinity. So more than 90% of the thymocytes produced by TCR that either do not bind to MHC peptide complex or bind to them with very low affinity. Yes, not with intermediate affinity, but with very low affinity. they die of apoptosis is 100% correct. So here statement 1 is incorrect, but statement 2 is true. So that means 4 is the right answer. So next question is with regards to the enzymes involved in the VDJ recombination. So first I will just brief out these enzymes what they have given in the options and their role in the VDJ recombination. So here I have shown the recombination between V2 and the J1 sequence. Actually it is the first the D and the J segments will join, then these two will join together to the V segments. But here I am showing the V2 and the J1 joining. The process and the enzymes involved are same in all cases. So first what happens is these RAG1 and RAG2 proteins, they will bind to the recognition signal sequence or RSS, these triangles ones which are flanking the VDJ genes. They will bind to these VDJ genes, that is RSS. The RAG proteins, they will bind to the RSS sequence flanking the V and the J gene segments. And this RAG protein, they will introduce a double strand break between the coding segments. Now there is the HMG protein that will help in the bending of DNA. That is what is shown here. That is a HMG protein which will assist in the bending of DNA and thereby helping RAG protein. Now after the RAG protein cleavage, what you will get is you will get two types of DNA ends. One is the coding regions, that is V2 and J1 having an hairpin. Okay, and the other is blunt signal ends, that is containing your RSS. Now these broken ends, these two are broken, that is double stranded break is introduced in the DNA. So they are joined together by the non-homologous end joining method. So you know this non-homologous end joining DNA repair pathway in all of the cells. You have the proteins Ku70, Ku80, then you have the DNA dependent protein kinase which gets phosphorylate, which will activate Artemis by phosphorylation. And Artemis, what it will do is it cut randomly in these hairpin loops, so it will result in the formation of overhangs in these coding segments, and these overhangs, they are filled by the terminal deoxynucleotidal transferase, and it adds the nucleotides in a random manner, as well as it doesn't require a template for the addition of nucleotides. And this is one of the reason for junctional diversity in these antibodies. Then there is the DNA polymerase mu and lambda that will fill any gaps. And finally the coding ends, they are these two, that is V2 and J1, they are ligated with the help of the DNA ligase 4. Okay. Now what is happening to these DNA segment with signal ends? So they are also just rapidly joined to form the signal joint which is lost from the cell. Okay, so that is what is happening in your VDJ recombination, and the various enzymes that are involved in this VDJ recombination. Okay. Now what is it that they have asked? Only 3 of the proteins implicated in VDJ recombination are unique to lymphocytes. One of the following proteins participates in the VDJ recombination of antibody genes and is unique to lymphocytes. Identify. So one of the protein out of these 4 what they have given here is unique for the VDJ recombination of antibody genes. It is not found anywhere else in the cell. So that protein is terminal deoxynucleotidal transferase. Okay. So this is the DNA polymerase that is exclusively expressed in your pre-B and pre-T lymphocytes. And I told you it adds nucleotides in a non-template dependent manner, and it is random addition of nucleotides, and it will result in junctional diversity in the antibody. So whereas Artemis, it is involved in non-homologous end joining pathway, so found in and non-homologous end joining pathway, it is for repairing DNA double-stranded breaks. So all other cells are also having that. DNA ligase for again a component of the non-homologous end joining repair pathway. Then HMG protein, that is also involved in bending of the DNA, and it is found in almost all the cells, whereas this first one, it is found mainly in the lymphocytes. So that is the answer. And next one, given below are two statements. One is labeled as assertion and the other is reason. Okay, now first statement, what they have given is type B T cells would sneak through the negative selection process in the thymus. So I don't know what is this type B T cells. I know only two type of T cell, that is TC and the TH. I am unsure about type B cells. So I have just removed type B from this statement, and when I read it, I found that it is somewhat correct. That is, T cells would sneak through negative selection process in the thymus. That is correct. T cells can escape the negative selection process in the thymus. And what is negative selection? I told you that that is for T cells, if they recognize self antigens bound to MHC molecules at a higher affinity, they will be undergoing the death signal or apoptosis. But those which bind with the very low with low affinity or around somewhat intermediate affinity, they will receive the live signal. That is, they can continue with the maturation process. Okay. Now they have given the reason as thymic medullary epithelial cells or dendritic cells are uniquely capable of forming MHC class II insulin peptide from exogenous sources. That is incorrect. It should be endogenous sources. Okay. Because see, for developing T cells that for negative selection, you usually subject the naive T lymphocyte to MHC molecule which is bound to a self antigen. So that means the antigen is of endogenous origin, not coming from outside. It is not any microbial antigen you are subjecting the T cell to recognize. Okay. It is the same self antigen. It is just, so negative selection is mainly to prevent autoimmunity, or else these T cells which have not undergone the negative selection, they will go and bind to all the cells in the body and they will start their action of throwing the cells from the body. That is not good. That will result in autoimmune conditions. So to prevent that, in thymic medullary epithelial cells, the T cells are subjected to negative selection process. So they recognize the difference between self and non-self antigens and thereby prevent autoimmunity. So when you are training the T cells in the thymus, you first give them the endogenous antigen, that is self antigen, not exogenous antigen. Okay. So here both this statement reason is incorrect. Okay, so statement assertion is correct. So A is correct but R is not correct. So answer is option 3. Then insect belongs to the arthropoda. So 4 is the answer. Then to hit origin of cancer, that is both the alleles need to be inactivated for a gene to be inactive was demonstrated and observed in the case of retinoblastoma, and this experiment was conducted by Alfred Knudsen. Okay. That is, if both the alleles of a tumor, suppose we take the case of tumor suppressor gene, if both the alleles, that is the one allele that you get from your father, the other allele that you get from your mother, if both are inactivated, then the cell can convert and transform, or it can become a cancerous cell. That is what two hit origin of cancer is telling, and this was first conducted in demonstrated in the case of retinoblastoma. Then telomerase enzyme is activated only in, so it is activated in stem cells as well as cancer cells, but not in differentiated somatic cells and muscle cells, which are also differentiated somatic cells. They are usually absent there or are found at very low levels. So A and C is the answer. Answer is option 4. Then next one, in a trisomic individual, the number of chromosomes is, so in trisomic individual the number of chromosome is 2n plus 1. 2n is the diploid chromosome number, and for humans it is 46, and if you take the case of Down syndrome as an example, they have 47 chromosomes. So 2n plus 1, that is 46 plus 1, it is 47. So Down syndrome individuals, they have are trisomic for the chromosome 21, or they have extra copy of chromosome number 21. So the answer is option 2, that is 2n plus 1. Then which of the following is an epigenetic factor that control gene expression at DNA level in eukaryotes? In majority of the cases, it is DNA methylation that is controlling the gene expression in eukaryotes. And epigenetic factor means it doesn't lead to changes in the DNA sequence. Okay. Now protein acetylation, if you take histone acetylation into consideration, that is also an epigenetic modification, but it is primarily affecting the histone modification, not your DNA. So they have asked here control of gene expression at DNA level, not at protein level. So it is DNA methylation, that is the answer. Then what is the chemical modification of a genome that is characterized as imprinting process? Now what is this genomic imprinting? So genomic imprinting, if I tell you, now you know that you inherit 2 copies of gene, that is one is from father and the other is from the mother. Now both the copies of the gene are usually active in the cell, but in some cases only one of the 2 copies is active. That okay. Now which copy is active, it will depend on whether it has come from the mother or whether it has come from the father. So in some cases the allele for this particular gene, it will be active only if it comes from the mother, but if it comes from the father, it will be inactive. That is called as imprinting. Now the genes for that undergo this genomic imprinting, they are often marked with the process called as methylation of DNA. Okay. So this methylation will help to identify which copy of the gene is inherited from the mother or whether it is inherited from the father. Okay, that is the principle behind this methylation of DNA in imprinting. So answer is option 2, that is methylation of DNA. Then next one, human telomeres have multiple repeats of sequence, that is TTAGGG, that is option 1. Then a man identified with the nervous system disease syndrome caused by mutation in mitochondrial DNA, that is the key word here, it is caused by mutation in the mitochondrial DNA. He married female without any such abnormality and is healthy. What is the chance of offspring getting the syndrome of father? It is 0. Why? Because mitochondrial DNA is inherited solely from the mother. The father doesn't contribute mitochondrial DNA to the embryo. Only the mother is contributing the mitochondrial DNA. Because mitochondrial inheritance is totally cytoplasmic inheritance or extra chromosomal inheritance. So the female is or the mother is not having any abnormality in the mitochondrial DNA and is healthy. So the offspring will always get the mitochondrial DNA from the mother, whether it is a boy or a girl. So the chance of the offspring getting the syndrome of the father is 0 only. And then next one, in mitochondrial mutation associated with the disease, the following 2 crossings were made. So the mutant is female here and the wild type is female in the case of the second cross. So in the first cross, the mutant is female and they have again told about mitochondrial mutation getting associated with the disease. So mitochondrial mutation, I just now told, it is solely uniparental. That is, only the female parent is contributing to the genotype of the offspring. Will contribute the mitochondria to the offspring. The father is not contributing any mitochondria to the offspring. So here if you see the first cross, mutant is female. So the progeny will be deceased because mitochondrial mutation. It is mitochondrial mutation. So the progeny will be deceased. So all progeny is mutant and deceased phenotype. That is correct. Now next one, female is wild type. So their progeny will not have any disease, even though the father or the male is mutant. So all progeny will be normal. So the answer is option 1. Then Mendel in his experiment did reciprocal crossing between 2 phenotype, plants of purple color with plants of white color. Now what is reciprocal crossing? So first condition, what you do is you take purple flowers as male and the white flower as female and you cross, you get the F1. Okay. Now in the second cross, what you do is inverse the sex of the parents. That is, you will take white flower as female and purple flower as male. Purple flower as female and white flower as male. That is reciprocal crossing. So reciprocal crossing produced F1 all purple colors. Okay. So by self crossing the F1, the F2 generation produced more purple colored plants and few white colored flowers. What observation would you infer? This is nothing but the normal Mendelian cross. So F1 all are purple color means F1 is the dominant phenotype. And in F2 generation only few are white colored, means white color is recessive phenotype. So the answer is option 1. Then next one, two plant genotypes. They have given the genotype there. A, small b, capital C, capital D, small d, capital E, small e was crossed with another parent of this genotype. Assume the gene assort independently. How many progeny plants are required to screen the whole recessive condition? So make separate crosses for each gene to get frequency of the genotype. That is the clue. So I have done as per the clue. So capital A small a into capital A. You need small a plant. So how many? What is the probability that you get a plant with small a is 1/4. Now small b with capital B small b, you get small b with half. Similarly for C, D, E you get 1/4, half and 1/4. Then what you do? Then probability of getting this particular genotype in the offspring is 1/256. That means 256 plants. Out of 256 plants, one will be of this particular genotype. So you have to screen either 256 or more progeny. So 2 is the answer. Then next one, metagenomic. So metagenomic, it is the study of genetic material from an entire community. That is usually the microbe community. And you take samples directly from the environment, that is soil or water or air. And the usual traditional method was you first isolate the individual species, then you culture, then you undergo genomic analysis. No, that is not done in metagenomics. Metagenomics means you will analyze all the DNA samples to understand the community's diversity, function, genetic relationship, all that. So answer is option 2, that is genomics, study of microbiome. Then next one, which of the following statement is correct for heterozygote advantage? So heterozygote advantage means you will have one allele from one parent and another allele from the other parent. So this is that is capital A and small a will be found in the heterozygote. Homozygous means capital A or small a. That is homozygote. Now they are telling that if the population is having this heterozygote condition, then that is advantageous for the population. Okay. Now read the statements. May lead to increased incidence of autosomal dominant disorders. No, because for autosomal dominant disorders you just need one copy of the diseased allele. It is dominant, so and it will cause the symptom. Now if this allele, if this capital A, if it is lethal, then the natural selection will act and the lethal gene will be removed out from the population, and it will the natural selection will try to keep the capital A gene in low condition, low incidence. Okay. Now heterozygote advantage, it is for autosomal recessive disorders. If you take the case of sickle cell trait and malaria resistance. So individual who are heterozygous for the sickle cell disease, that is they have sickle cell disease is a recessive condition. So they have small a which is sickle cell trait and capital A which is the normal allele. So they will have increased fitness and they over the homozygous conditions, and especially in regions where malaria is very prominent. So those with the heterozygotes, condition, they will not suffer from severe forms of malaria. Okay. So the heterozygous individuals will be selected for by the natural selection, and they will be passed on to the next generation at a high frequency. So not autosomal dominant disorders, it is autosomal recessive disorders. Now heterozygous advantage is very unlikely to be traced to founder effect. As per me, that statement also is correct, but as per your answer key, the answer is option 4. So it is left up to you to choose. Now what is founder effect means? A new population is created when by a small number of individuals leading to different allele frequencies when compared to the original population. Now because of this, it will reduce the diversity in the the founder effect will reduce the diversity in the population. Okay. But founder effect, it cannot be traced to heterozygous advantage cannot, or it is very unlikely that it can be told that it is caused by founder effect. Yes, because heterozygous advantage it is the favoring of the heterozygous genotype over the homozygous condition. Just like in sickle cell trait in malaria prone regions, the heterozygous will provide resistance to malaria, and they will be selected for by natural selection than the homozygous one. And heterozygous, it will favor what is that? Or I can say it will maintain genetic variation and fitness of the heterozygous condition. So it is very unlikely that the heterozygous advantage is traced to founder effect is also correct. Now does not distort Hardy-Weinberg equilibrium, that is also not correct. Now Hardy-Weinberg equilibrium, the key assumption is there is no natural selection, no mutation, no migration. It happens in large population and there is random mating. Now this heterozygous advantage is also a type of natural selection. That is the heterozygous genotype which is having higher fitness than the homozygous genotypes is selected by nature. So it is a type of natural selection. Whereas Hardy-Weinberg equilibrium, it says that no natural selection is working on the population. So it does not distort Hardy-Weinberg equilibrium. Is wrong. Then may explain the worldwide distribution of sickle cell disease and malaria. Yes. So individual with normal hemoglobin, they are susceptible to malaria. That is capital A, capital A. And individual with recessive condition, that is small a, they have anemia and various other health problems and can be life threatening also. Whereas those with capital A, small a, they carry one normal allele and the other sickle cell allele. They do not suffer from severe symptoms of malaria under normal condition and are usually protected from malaria. So it is a selective advantage or for the heterozygous condition in malaria prone regions. So 4 is also the answer. Then next one, which of the following is incorrect for mitochondrial disorders? Now all follow MAT matrilinear inheritance. Matrilinear means maternal inheritance. Now, do all mitochondrial disorders follow maternal inheritance? No. Mitochondrial disorders means mutations in the mitochondrial DNA that are follow strictly maternal inheritance. But majority of the proteins that function in mitochondria are encoded by the nuclear genes. So mutations in nuclear genes can cause mitochondrial disorders also. And these mutations which arise in the nuclear DNA, they follow the Mendelian inheritance pattern, not the matrilinear or the maternal inheritance pattern. So this statement is not correct, and it is the answer for the question. Then less than 50 gene products from mitochondrial genome. Yes, because the mitochondrial genome, it encodes somewhere approximately around 37 gene products. So that is less than 50. So that statement is also correct, but not the right answer. Then retinal pigmentation and diabetes both could feature yes, mitochondrial disorders. Yes, that is also correct, because severe mitochondrial disorders can feature both retinal pigmentation and diabetes mellitus, but that is not the answer for the question. Then Leigh disease is a mitochondrial condition. Yes, that is also correct. And it is caused by gene mutation that impair the body's ability to produce energy in the mitochondria. So that is also not the correct answer for the question. Then next one, which of the following options are correct for immunology? Okay. So the immunoglobulin molecule is made of 6 polypeptide chains, which is not correct. It is of 4 polypeptide chains, that is 2 heavy chains and 2 light chains. So that makes option A gone. That means option 2 also gone, option 3 also gone. Then next one, the DNA encoding the K light chain contain 4 distinct regions. So that is correct. Then close relatives make the best organ donor because they are likely to share same complement, not complement, it is HLA haplotypes. HLA. So close relatives make best organ donors primarily because they are likely to share the same human leukocyte antigen or the HLA haplotype, not the complement. Complement means your C2, C4, factor B, all these things. No. Even though these C2, C4 factor B proteins, they are also located within the MHC region. But they are not the ones for organ donor you see for the HLA one. Okay. So C is also not correct. Then the diversity of T cell surface antigen receptor can be compared with the process of immunoglobulin diversity. Now T cell surface antigen receptor is nothing but your T cell receptor. Now T cell receptor and your immunoglobulin diversity both are generated through the VDJ recombination that randomly joins the V, D and J segments during the development of the B and the T cells. So that statement is correct. So B and D is correct. So 1 is the right answer. Then next one. Which of the following statements are correct for sickle cell disease? So HbS, that is the hemoglobin of sickle cell disease, differs from the normal hemoglobin Hba by single amino acid substitution. That is 100% correct, because here the valine replaces the glutamic acid at the 6th position of the beta globin chain. Okay, so that's correct. So that means 4 gone. Then the sickling effect of red blood corpuscles is the result of abnormal hemoglobin binding with the red cell membrane. No. Now this the sickling effect, abnormal hemoglobin means the sickle cell hemoglobin. Okay. Now if this is sickle cell hemoglobin, it releases oxygen and become deoxygenated. Then these abnormal hemoglobin molecules, they can join together to form a stiff chain, rigid and stiff chain inside the red blood cells. Now this rigid chains will give the characteristic sickle shape to the red blood corpuscles. So the sickling effect of red blood corpuscle is the result of abnormal hemoglobin binding to the red cell membrane is not correct. It is abnormal hemoglobin binding to itself under deoxygenated condition. So B is wrong. So that makes 1 also wrong. Then next one, splenic infarction occurs, but this has little clinical consequence. That is incorrect. It can have even severe and life-threatening complications. So C is also incorrect. So 3 also gone. That makes option 2. That is A and D are the correct. Life-threatening thrombosis can occur because of sickle cell disease. That is also correct. So that makes what is that? A and D as the right answers. Then which of the following chemicals are used in the preparation of metaphase chromosome for analysis of light microscopy? So phytohemagglutinin, it is used to stimulate the peripheral blood lymphocytes to begin division in culture. For that you use, yes, you have to first divide the cells to reach the metaphase chromosome. So that is required. Then colchicine, if you see, it will arrest the cells in the metaphase stage by disrupting the formation of mitotic spindle fibers. So that is also required. Then hypotonic saline means water will enter the cell via osmosis, swelling of the cells. This swelling will separate the chromosome from each other and prevent them from clumping, and you can spread the chromosomes on a slide and use it for your light microscopy analysis. So D is also correct, but what about quinacrine? It is a fluorescent dye. So light microscopy cannot be used. You have to go for the fluorescence microscope. But if you want to see with the light microscopy, you can go for the G staining or the Giemsa staining. So wherever B comes, that option is wrong. So A, C, D, that is 3 is the answer. Then next one, in lionization of X chromosome inactivation, all the genes of one X chromosome are switched off. All the genes are not switched off. Some genes located in the pseudo-autosomal regions of the X chromosome, they escape the inactivation process and they remain active in both the X chromosomes. Okay, so that makes statement 1 as wrong. Then next one, in most phenotypic males with the karyotype 46XX, the SRY is present and found on one of the X chromosome. Yes, that is correct. This is because the SRY gene, which is located in the Y chromosome, is translocated from Y chromosome to the X chromosome condition. So these people, they will be phenotypically, they will be males and their karyotype will be 46XX instead of 46XY. Okay, so that statement is correct. So statement 1 is false and statement 2 is true. That makes option 4 as the answer. Then which of the methods can be used for gene therapy of cancer? Inhibition of fusion proteins. Yes, they can be done because some of the fusion proteins, they will activate the oncogenes. Oncogenes means they will promote, activation of oncogenes means they will promote cancer growth. So that is correct. Then comes the what is that? Stimulation of immune system. Yes, that also can be done because if you stimulate the immune system, then the immune system will recognize and attack these cancer cells. So that also can be done by gene therapy. Then increased expression of angiogenic factors. Now angiogenic factors, they are the ones that promote tumor. And angiogenic factor means they help in the formation of new blood vessels. So like they will promote tumor. So increased expression of angiogenic factors means you are promoting tumor, not treating the tumor. So C cannot be correct. Then RNA interference means you can silence specific genes involved in the cancer progression. So D is also correct. So A, B, D are correct. That makes option 2 as the answer. Then next one. For a pre-implantation genetic diagnosis, which of the following does not take place? Okay. Now what is pre-implantation genetic diagnosis? Is usually done in in vitro fertilization technique and they will test the embryos for genetic disorders after conducting the in vitro fertilization procedure, and only after testing these embryos for genetic disorders they will decide whether they have to be implanted in the uterus. So those which are having some disease carriers, that is means those embryos which are having some genetic disorders, they will not be implanted into the uterus. Only the healthy ones without any genetic disorders, they will be implanted into the uterus. So that is what pre-implantation diagnosis is done. So here they say the use of fluorescence in situ hybridization to detect an unbalanced chromosomal abnormality. Yes, that is done for pre-implantation genetic diagnosis. So through this in situ hybridization technique, you can identify if there are too many chromosomes or whether there are too less chromosomes or whether there is any structural abnormality in the chromosomes, like even translocations. Okay, so all these can be studied with the fluorescence in situ hybridization before implanting the embryo into the uterus. So, but that is not our answer. They have asked does not take place. Now next one, fetal sex determination of embryo in sex-linked disease. Yes, that is also taking place in pre-implantation genetic diagnosis. Okay. So here you determine the sex of the embryo. If the parents are carriers for a sex-linked disease, so if it is X-linked disease, naturally if the embryo is of the male, then it will not be transplanted into the uterus of the mother. So here because X-linked disease recessive condition means males will have more chance of inheriting the X-linked disease. Okay. So here fetal sex determination is done to see if the embryos are is a male, if it is X-linked recessive condition. Okay. If it is male and having the X-linked disease, then it will not be implanted into the uterus. So that is also done. Then testing each of the cells of the embryo for specific mutation. Now each and every cell, single cell of the embryo is not tested. A few cells are taken and tested, and they will check for any specific mutation. Okay. Not each and every cells are selected. So that is the right answer they have asked, does not take place. So each of the cells is not done in PGD. In vitro fertilization, yes, that is also the for pre-implantation genetic diagnosis.